for inviting me. I'm thoroughly enjoying this conference. And yesterday when I went back to my room in this old building, so all of a sudden I realized that maybe Charles Darwin stayed in that room sometime in the past. I found this very inspiring. Okay, so my laboratory is interested in relation between genome stability and aging process, and also in using comparative approach to understand aging. And this is this is the project that I'm going to present to you today. So what is comparative biology? Basically, comparative biology of aging. Evolution has created species with dramatically diverse aging rates. And I'm not talking only about differences of Drosophila versus whale, but there are some species that are quite related to each other, and still they can have up to tenfold differences in lifespan. For example, here you can compare a rat to a squirrel, which are both rodents, and the rat typically lives, 4 years, or maybe even less, while eastern gray squirrel, and that's American squirrel, lives up to 24 years, so people are often surprised that these creatures can live so long. So if we could identify what makes this species lifespan so different, so then perhaps we could answer many questions that are also related to human aging. My laboratory focuses primarily on the rodent clade, and the reason we really like rodents, so it seems that this clade has been there ideally suited for comparative aging studies. And the reason is that within this group we have species with really ranging from rather short-lived to quite long-lived species. So for example, well, you're familiar with mouse, laboratory mouse and rat that are pretty short-lived even for a mammal, small mammal. And then we have some very long-lived species, so naked mole rat that lives up to 30 years, squirrel that I already mentioned, there is a beaver, porcupine that all live over 20 years, and then there is anything in between. And if we look at rodent phylogeny, so here I mark with an asterisk those species that have extremely slow aging rates and live over 20 years, and you can see right away that they are situated on different phylogenetic branches, meaning that within rodents, slow aging has evolved independently at least 4 times, meaning that it comes extremely handy when doing comparative studies, because we can easily differentiate between those mechanisms that are evolved as adaptation to slow aging versus something that just has to do with those species being related or belonging to the same group. So today I'm going to describe, and so another interest of our lab is cancer and the various tumor suppressor mechanisms. So I will talk about one mechanism about telomere biology, which is something that's extremely relevant to both cancer and aging. And Maria Blasco gave a very nice introduction to telomere, so I won't talk about telomere is, but I'll just briefly mention the phenomenon of replicative senescence because it's going to be quite relevant to this topic. So if you take human cells and put them in culture, so primary human cells are not immortal, and so for example, if we are talking about fibroblasts, they will divide a certain number of times, and then they arrest proliferation and enter so-called senescent state. So this senescent state contributes to the aging process, especially aging of proliferative tissues, as already been mentioned in the previous talk. However, of course, this mechanism didn't evolve to shorten our lifespan, and the primary function of this is to be an anti-cancer mechanism. So it's a powerful tumor suppressor. And very early on, when replicative senescence was discovered in human cells, it was noticed that there is a dramatic difference between human and mouse telomere biology, and actually this sort of paradox was what attracted our attention in the first place when we started on this project. So if you compare human to a mouse in terms of telomere biology, so in humans, as I already said, telomerase is repressed in most somatic tissues. Telomere length is relatively short, and if you put human fibroblasts in culture, they display replicative senescence, and that's true for other cell types as well. While in mouse, telomerase is active in most somatic tissues, I mean not in all of them, but in majority. Telomeres are much longer than in humans, and mouse fibroblasts, if they are cultured under low oxygen concentration, they do not display replicative senescence. So now, perhaps in part as a result of these different strategies, mice are much more prone to cancer than humans. So in some mouse strains, up to 95% of animals die of cancer, while in the human population, cancer mortality is about 25%. And so in general, the way people used to explain this difference was because, well, humans are much longer lived. And then, well, perhaps they need all these mechanisms in order to protect themselves from cancer. And in case of a mouse, maybe, well, that's a trade-off. Mice do not need to protect themselves from cancer as much as humans do, because in the wild, they never get to live long enough to develop cancer. They're usually being preyed upon by, there are lots of animals that prey on mice in the wild. So they live maybe 6 months at most. And cancer is really not a big issue for them. So what we were interested in is to test whether indeed evolution of replicative senescence and repression of telomerase is related to lifespan, if all species evolve this tumor suppressor mechanism. And when we started working on it, there has been really little information about species beyond human and mouse, so because primarily cancer research focuses on these two important species, human and mouse. So in order to test this hypothesis, what we had to do was to take all those rodents that I showed you in the picture before and test telomerase activity in their tissues. So in order to do that, so we obtained multiple individuals of each of 15 rodent species and measured telomerase activity in a set of tissues in each animal. So we used the trap assay, which is quite sensitive assay for telomerase activity. So briefly, you take cell extract and you give radiolabeled primer. So if there is telomerase in the extract, it will extend the primer in the multiples of 6 nucleotides. And here are some examples of trap assays that we observed. So here is our positive control, which is a human cancer cell line. And so you can see this characteristic ladder corresponding to high telomerase activity in the extract. And so this is a panel of mouse tissues. And you can see that there is quite a bit of telomerase activity in the number of tissues. Some tissues have a bit lower activity, but it's still present. So now let's look at other species. And here I just give you an example of 3 longer-lived species among rodents. So first we looked at the beaver. It lives over 20 years. And we detected almost no telomerase activity in beaver tissues, well, except for the skin, so there is low activity in the skin. And if we were to test human tissues for telomerase activity, that would be about the picture that you expect to see, because there is some low activity, telomerase activity in human skin due to stem cells found in the skin, but otherwise other tissues have activity that's just below detection for this conventional trap assay. So when we saw that, we thought, oh, well, it fits perfectly the hypothesis. Now, beavers are long-lived, they have replicative senescence like human, very nice. But then we looked at a squirrel, another animal that lives over 20 years, and all of a sudden we realized, well, it all falls apart. Because squirrels have extremely high telomerase activity in all the tissues we tested. Even squirrel heart has high telomerase activity. So then we thought, oh, well, it's not what we think. Next, we look at naked mole rat, and again, similar situation. There is telomerase activity everywhere we look. So then what we've done, we quantified telomerase activity